Hello, everyone. So today we're going to be talking about our biology EOC review lesson number 2. So in this particular review lesson, so we'll be talking about a lot of standards. So I'm going to go over one by one. And the first one we're going to talk about is why are cells so small? That is the question that we're going to address that. And the answer is simple. The smaller the cell is, they are going to be more efficient. Why? Because, and here is the mathematical modeling that you can see that here, and there are three sets of cube, and this is right here is one by one. It's a very small, and you can see that one big cube right here, which is all the sides are in a unit of 5. And here is another one with the same volume right here. Is these two are same volume, but you can see this is made up of many number of cells. What I want you to notice here is, so here the total surface area for this particular cell is 6, and this particular cell right here is a larger one. It is 150, and you can see here this one is made up of many number of cells, and the volume remains the same, and this has a higher surface area, which is 750. And you can see that the total volume in these two stay the same. It is 125, 125 for the volume, and here is 1. And I want you to look at the ratio. For this one is 6, for this one is 1.2, and look what's happening here. And having this big size, and the surface area to volume ratio is 6 here. So which is really a great advantage, having this high number surface to volume ratio. And you can see that the surface area is high right here in this particular unit, so that the materials can really absorb into the cells, and also it can release out very efficiently. And that's the reason the whole entire human body is made up of billions of cells instead of one cell. So in this particular slide, we are going to quickly review. It's from yesterday's lesson, different type of organelles, and you know I'm going to quickly review that. Nucleus is the control center of the cell. It has the DNA. Ribosomes are responsible for making proteins, and to reach the messenger RNA come from the nucleus. Golgi apparatus are responsible for shipping and packaging and send the vesicles out of the cell. Endoplasmic reticulum, it's mainly responsible for synthesizing cellular materials. It has smooth ER and rough ER based on the ribosomes are present or not. Nucleolus is the structure that makes the ribosomal RNA. It helps in making ribosomes. Mitochondria is the powerhouse of the cell. It performs the cellular respiration. Vacuoles are responsible for storage bags. It stores the minerals and water. And vesicles are responsible for transporting the material out of the cell by exocytosis. And cytoskeleton is it's pretty much the protein fibers inside the cell. It is like a skeleton in our body. It gives the physical support. Flagella, cilia, and pili, these are the protein threads, filaments. It helps the cell to move. And these green, the one which is shaded green, are only for plants, which is cell wall, chloroplast, and central vacuole. And these two are only for animals, which is lysosomes and centrosome. And the lysosomes contain enzymes that help to break down unwanted materials. And also the centrosomes responsible for, it containing centrioles. It helps in cell division. So the next lesson we're going to quickly review is properties of water. And water is an universal solvent. It is the reason why this biosphere is filled with water. It's because of some special properties. The first one is water is a polar compound. What does that mean? Look at this water molecule right here. This is one water molecule. This is another water molecule. This is another one. This is another one. This is another one. And look at this oxygen atom right here. Oxygen atom is attached to the hydrogen atom within the water molecule by covalent bond. But believe it or not, this oxygen is so highly electronegative. It loves electrons, so it attracts from the neighbor hydrogen. So it gains some negative charge, and the hydrogen gains some positive charge. So because of this positive and negative charge between the water molecule, it attracts each other and forms the hydrogen bond. So because of these charges, we call water is polar. It helps the water to act as a best solvent. It pretty much dissolves all the material, including salt and sugars. Another property of the water is the high heat capacity. That means you have to really give a large amount of heat to change the temperature of the water. That's why our body temperature doesn't change that quickly as fast, because our body is filled with water. In the same way, the large water bodies, the animals are protected because of this a property as well. Next is the water has a high heat of vaporization. That means if you want to vaporize water, you have to really heat it up compared to other liquids. Um, so that is why we call water has high heat of vaporization. Another property is water has a cohesive and adhesive property. That means water attracts water. You can see that by hydrogen bond, they attract water and they stay together. That's why water can climb up through the stem to reach the tip of the tall plant's upper canopy. Also, there's a transpiration pull helps to climb up the water through the tubes as well, which we call xylem. Adhesion is the property of the water. It attached to the different surfaces. Water attracts many different surfaces. So these properties helps the water to climb through the xylem or the pipes in which the water transport in different parts of the plant. The last property is water is less dense as a solid. That's why ice floats on the surface of water. When you dispense some ice from the ice machine and then fill it up with the water or iced tea, you can see that the water floats on the surface of water, surface of the water or the iced tea, because the ice floats on the water because it is less dense. Do you know why it happened? When you freeze ice, these hydrogen bonds push the water molecules away and forming a crystal lattice that increases the volume of the water molecule, the whole as a crystal. And so increasing the volume decreases the density, and that's why it floats on the surface of the water. Now I'm coming to the last section of today's review lesson, which is macromolecule. There are four different type of macromolecule that makes our living system. First one is carbohydrates, second one is lipids, third one is proteins, and fourth one is nucleic acids. So what do you see here is the carbohydrate. Now our carbohydrates are hydrates of carbon. It is made up of carbon, hydrogen, and oxygen. So I pronounce this as CHO. And you can see this chart here. It tells you different properties of this macromolecule carbohydrates. Carbohydrates is made up of carbon, hydrogen, and oxygen at 1:1 ratio. The monomer or the single units in which carbohydrates are made up of is monosaccharides. And these monosaccharides are example simple sugars like a glucose. Disaccharides are another type of carbohydrates. It is made up of two monosaccharides, and polysaccharides are made up of many monosaccharides. Example, starch, glycogen, cellulose, chitin. These are examples for carbohydrate, which are polysaccharide. Disaccharide is the sucrose, which is our table sugar, which we add in tea and coffee, and monosaccharides are glucose, fructose, and galactose. What's the primary function of carbohydrates? It really gives us energy. That's why we eat a lot of carbs, is our main part of the diet, so it gives us energy. And of course, the cellulose is the part, and chitin in insect. The chitin, their exoskeleton is made up of chitin, so that is the part of the structure. The next macromolecule is again made up of carbon, hydrogen, and oxygen, which is lipid. Lipids are nothing but the fats and oil. It is usually their ratio in which the carbon hydrogen ratio is 2:1 or even greater than that. The monomer of the lipids is the fatty acid and glycerol. So there are different type of lipids. The first one is saturated. Saturated is usually they are solid at room temperature, which is animal fat. There is no double bond. All the carbon hydrogen chemical bonds are single bonds. And another one is monounsaturated, polyunsaturated. Then phospholipids are the type of lipids that is found in our cell membrane. So another example for the lipid is the steroid. Steroid is it has a molecular ring structure. Another one is wax. Wax is found in either honeybee or on the surface of the leaf. So those are also example for lipids. And you can see here wax as example bee wax, and the main use is to repel water and also on the surface of the leaf. Steroid example, you can see cholesterol, testosterone, progesterone. These are hormones which are chemical messengers, and phospholipid is a cell membrane. It protects the cell. And this is olive oil is an example of unsaturated fatty acid, which is oil. All the plant oil is pretty much unsaturated fatty acid, which is a good fat. The next macromolecule is CHON, is made up of CHON. It is carbon, hydrogen, oxygen, nitrogen, which is protein. And you can see that it is the monomer for the protein is amino acid. And when two amino acids are linked together, we call them dipeptide. And many amino acids linked together, we call polypeptide. Example for the proteins, enzymes in the body, stomach enzymes, salivary enzyme, muscles are proteins, steak, remember that, and skin has a lot of proteins, and some hormones like insulin are proteins. So main functions of the proteins are really essential: storage in chicken, egg, chemical messengers or signals, structural protein, which is muscle, contractile protein, which is cilia and flagella, defensive proteins, which is antibodies, enzymes, all enzymes in our body are proteins, transport proteins, which is in the cell membrane, move the molecule in and out of the cell, and receptor protein in the cell membrane as well. Last macromolecule, which is nucleic acids, made up of carbon, hydrogen, oxygen, nitrogen, and phosphorus. And you can see here macromolecule, example nucleic acids, example is DNA and RNA, excuse me. Um, so the main functional group we can see here, CHONP, CHONP, that's the main elements which is made up of. The monomer is nucleotide. A nucleotide in DNA is made up of a ribose sugar, phosphate, and a base. Main function of the nucleic acid is it store and transmit genetic information, which is the DNA and RNA, you know that. The last section in this video, which we are going to talk about, is the scientific method. And you know that you are doing science fair experiment or some type of lab activity you have done already. So the first thing is you ask a question: what experiment I am going to do? And then you research about the topic, then you come up with the hypothesis in if-then format, then you design your own experiment, and you follow the procedure to do the test the hypothesis, then you collect the data, and you analyze the data, and you come up with the conclusion that whether, hey, did that work, did that go according to my prediction. So that is the steps in the scientific method. And as you can see here, in this experiment, different amount of water has been given to these potted plants. And so the different amount of water that I add, which is called the independent variable, that is what causing in different ways for the plants to grow. And you can see that this plant grow really well, which is the normal water. And you over poured the water, it died, and the less water, it really did not survive well enough. So that is the variable affected by the change we call the dependent variable. You can see the number of leaves on this plant that can be used as a collecting the data, which is the dependent variable. Now when you plot these independent and dependent variable on a graph, always remember in this experiment, independent variable, which is the time, goes on the y-axis, excuse me, x-axis, which is the horizontal axis right here, and the y-axis is the speed in meter per second. This is another experiment. I just put this data here to make sure you understand. So if I'm making a graph out of this one, the amount of water that you add goes on the x-axis, and also the number of leaves on the plant goes on the y-axis. And another thing, one last thing I would like to mention is the experimental group and a control group. And if you want to create another control group for this experiment, you better don't add any water to the plant. So that is going to be a control group, and then you can test whether adding water is going to be a good thing for the plants or not. You can test it, which is going to be the independent water, so independent variable, which is amount of water that you're adding to the experimental group and not adding to the control group. And dependent variable is, in this experiment here, how many sprouts you get. So, and again, independent variable goes on the x-axis, dependent variable goes on the y-axis. And another way I would explain is independent variable is the cost, which is the water. Dependent variable is the growth of the seeds, which is we count as number of sprouts coming in. So if you have any questions, please let me know or email me. And I hope this is helpful to you. Thank you.